Good afternoon and welcome. I'm Kathleen Brackett from Kudo Biotechnology, and I'm excited for you all to be here for Kudo's clinical batch manufacturing and process development case study titled A High-Quality Delivery of mRNA LNP with an Unbeatable Timeline. I would like to introduce the presenters today. Dr. Kelvin Chan is Director of Commercial Strategy and Business Development at Kudo Bio. He is a globally experienced healthcare investor, operator, and formerly Director of Business Development at ABioX, a CBC Group biotech incubator. Dr. Chan holds a PhD in organic chemistry from Scripps Research. We also have with us today Dr. Atsuko Sangria, Director of Drug Substance Process Development. I will let Kelvin give you more information on Dr. Sangria and also our company overview in a moment. But before we get started, I wanted to let you know we will have a live Q&A session at the end, so feel free to submit your questions during the presentation from the box on your home screen. Also, we will have a link to a survey under the Resources tab that we hope you will fill out. For everyone who participates, you will be eligible for a $25 Amazon gift card. So without further ado, I'm going to turn things over to Kelvin. Kelvin?
Thanks, Kathleen, for the introduction. Hello, everyone, and thanks for spending your time today with us. My name is Kelvin, and I'd like to spend just a few minutes to introduce you to Kudo Bio. Kudo is a global mRNA CDMO, providing a complete and world-class suite of solutions for mRNA all under one roof. We offer pDNA, mRNA, LNP, and fill-finish GMP manufacturing on our end-to-end platform, as well as process development and analytical development. We have a track record of producing multiple batches of GMP products that have already been used in global clinical trials. Kudo has a state-of-the-art clinical GMP manufacturing facility and MSAT labs in Shanghai, a brand new process science center of excellence in Needham, Mass. That Atsuko will tell you more about, and we also have a presence in Singapore. Our team brings decades of experience working with complex biologics from preclinical all the way through to commercial stages, both at CDMOs as well as at innovative companies. We have been successful at international tech transfer, so we are confident to help you move your programs forward. As mentioned in the previous slide, Kudo has presence around the world. Only this week, we opened our Process Science Center of Excellence in Needham, Mass. That offer both process development and analytical development capabilities, so you are among the first to know. Our manufacturing hub and MSAT labs are located in Shanghai, China, and is fully staffed, fully operational, and fully qualified, certified to ISO 9001 standards. Our GMP facility spans over 57,000 square feet or 5,300 square meters, and is designed in accordance with cGMP and global regulatory guidelines. Our facility features 2 plasmid lines, 3 mRNA lines, 2 LNP bulk lines, and 1 drug product fill and finish line. It can cater to manufacturing needs for preclinical through phase 2 clinical trials, with capacity to manufacture up to 40 million vials per year for phase 3 and commercial manufacturing. Our cutting-edge equipment for manufacturing and quality is customized for mRNA and LNP drug development as seen here. I'd like to highlight the importance of having all parts of the manufacturing workflow from pDNA to fill finish all under one roof. We know that there are providers who cater to specific segments such as mRNA IVT or LNP fill finish. However, working with multiple CDMOs and suppliers means balancing slot availability, multiple handoffs to different project managers, which is inefficient and increases risk unnecessarily. At Kudo, we're able to do all at a single site and can accelerate the entire process in an integrated and truly end-to-end manner. This streamlined process allows Kudo to deliver our differentiation in performance, speed, and quality for CMC. Here you can see some metrics that we benchmark ourselves to, from capping efficiency to mRNA recovery and LNP encapsulation at or exceeding industry standards. We can deliver GMP products from sequence to mRNA LNP in 6.5 months and in 3.5 months if starting from pDNA. Quality is of utmost importance to us, as we know how critical this is, especially when products enter the clinic. Kudo is in full compliance with global regulatory requirements, including key parameters such as sterility, bioburden, endotoxin levels, particle size, and many more. This slide shows the production batch sizes that we typically offer for GMP and non-GMP needs. For pDNA, we can do up to 2 liters for non-GMP and up to 200 liters for GMP. For the IVT process, Kudo manufactures up to 100 ml for non-GMP and up to 2-liter reaction vessel size for the GMP process, which means a batch size of 1.2 liters. For LNP, this is pretty similar to IVT, yielding up to 6 grams for GMP. We have the capability to do fill finish up to 2,600 vials per batch, which is rate limited by visual inspection. Now, each of these process units are modular, so you can choose to start at any stage. For example, we can start at linearized pDNA and make mRNA perhaps without LNP, and we can ship material to you in bulk or in your shipment vial of choice. Or we can do the entire process train from pDNA through to the fill-finished drug product in bulk or in vials. We are flexible and highly customizable to your needs. Kudo's quality is delivered at the highest levels possible through our robust quality management system, which encompasses all the necessary process elements from personnel to environment to QC and product release, as well as the QA elements such as change control, quality reviews, internal audits, and much more. Our QMS has been audited by an independent ex-FDA auditor who recognized our system at a commercial quality level, which significantly surpasses the requirements for CMC. We also offer modern IT infrastructure to enable remote tech transfer and inspections for QA oversight from our clients in real time. A value-add capability of Kudo's offerings is providing options for sources of high-value raw materials. We have pre-audited and pre-qualified suppliers with complete data packages and provide you with alternatives to raw materials, including enzymes such as T7 RNA polymerases, capping reagents, nucleotides, and lipids. These give you options that can help ease cost burden or any IP burden that comes with other raw materials. Importantly, we also know these raw materials work well in our hands and on our platform. I hope you now have a better understanding of Kudo's capabilities, which will give you more context on the case study. I'd love to introduce my colleague, Dr. Atsuko Sangria, who will be the speaker for this next section. Dr. Atsuko Sangria is the Director of Drug Substance Process Development at Kudo Bio. She has 16 years of research experience in molecular biology and vaccine development. Prior to joining Kudo, Dr. Sangria led the mRNA drug substance process development, scale-up, and tech transfer at RVAC Medicines, an mRNA product company. Before that, she was an expert scientist at GSK Vaccines, where she played a vital role in mRNA vaccine platform development. She also has preclinical and process development experience in biologic products from her past role at VLP Therapeutics and postdoctoral training at the National Institute of Health. Dr. Sangria obtained her PhD in pharmaceutical sciences from Kitasato University in Japan. Atsuko, over to you. Thanks.
Thank you for the introduction, Kelvin. So I'm Atsuko Sangria, Director of Drug Substance Process Development at Kudo Biotechnology. Today, I'm going to present a case study about how Kudo's streamlined approach provided the solution for our client. The drug developers are always seeking a way to get to the clinic as quickly as possible, and during the pandemic, it was demonstrated that RNA technology can bring products to the market in record time if it's done well, so development timelines accelerated and it became new normal. Our client, RVAC Medicines, is a global clinical stage RNA platform company based in Boston with a pipeline of innovative products that covers a range of disease areas. Arvac Medicines did not have an established internal group for process development, so their CMC vice president reached out to Kudo for both process development and manufacturing projects. Arvac Medicines had two key objectives. First, they had an ongoing clinical trial of RNA COVID vaccine, and they urgently needed a resupply of clinical trial material. The second objective was for their future programs. Arvac Medicines was looking to evaluate alternative critical material suppliers to avoid supply chain delays. Arvac Medicines needed a reliable, efficient CDMO with process development capabilities to complete both GMP and process development requests. For the first objective, the challenge came from their manufacturing campaign involving 3 different CDMOs. This is a pretty standard approach for RNA products. The template DNA is manufactured at the DNA company, released and shipped to the second CDMO, which is the RNA manufacturer. They produce drug substance, release, and transfer it to another site, and then at this CDMO, the RNA is formulated in LNP and then released and shipped to the clinical trial site. So when Arvac Medicines needed to resupply the clinical material, CTM, it was challenging to book the slot at each CDMO and coordinate activities at different sites under the tight timeline. Also, it could be very expensive considering the project management cost at 3 CDMOs, and communicating with each one of them is very time consuming. You want to perform stability studies, but at 3 sites, and these costs adds up. On the other hand, Kudo has all 3 functions under one roof, so this streamlined approach solved these challenges for Arvac Medicines. So here is the example process flow of RNA product formulated in lipid nanoparticles. The first step is DNA linearization. Then the RNA is produced by in vitro transcription, IVT, and capping reaction. Then the RNA is purified using TFF, tangential flow filtration, and oligo-DT chromatography, and if needed, extrapolation steps. And then final drug substance goes to the LNP formulation. Then the bulk drug product is filtered and filled to the final containers. Kudo can perform the whole process at a single manufacturing location, which is very efficient because you will face less challenges in timeline, logistics, and so on. Smooth and efficient tech transfer is critical to a successful GMP campaign. So in this case, 2 technology transfers were required. So one for drug substance process and then one for LNP drug product process. We maintained frequent, open, and transparent communication between donor and acceptor sites. And again, having both DPs, drug substance and drug product facilities under one roof helps since it's one team. Then another important element for any tech transfer is analytical method transfer and qualification, which can be rate limiting step. So Kudo has very robust in-house analytical team, worked very closely from the beginning with the process team to make sure there's no gaps, no surprises. This is a workflow for the Arvac Medicines GMP campaign at Kudo. So their COVID vaccine, RVM-V001, is a linear messenger RNA, capped and poly-A tailed. Their IVT condition was based on the DOE study performed during the previous GMP campaign. So the initial DOE study was conducted with 4 varying factors: temperature and concentrations of NTP, magnesium, and T7 RNA polymerase. Kudo took it from here. Based on the result of this DOE study, we performed another DOE study with less conditions. NTP, magnesium, T7 RNA polymerase concentration was evaluated. Then as a response, we try to maximize the RNA yield and integrity while reducing the dsRNA impurity. So as you can see in this graph here, we determined several good conditions. So IVT yield is shown here as blue bars, and these pink dots are integrity of each IVT product. As you can see here, conditions 8 and 10 were determined as high quality, high yield conditions. So we chose 3 different conditions and then did the reproducibility run in small scale to confirm the conditions. After that, we did the 2 pilot scale runs with scale down process with downstream purification. 2 independent batches were made. We confirmed everything looked good here, proceeded to the full-scale GMP run for the actual clinical trial material production. So here we show the summary of our findings from the DOE studies and the results of confirmation IVT. As previously reported, magnesium concentration had a significant impact on RNA yield. Higher temperature, like during the IVT reaction, resulted in low dsRNA, but RNA integrity was negatively impacted. We selected 3 conditions and performed confirmation runs, and as you can see here, this is the representative image of fragment analyzer results. You can see like a nice single peak in each condition. So from the integrity and yield, we chose this condition A as the best condition. After the confirmation IVT and pilot scale runs with scale down model with purification, Kudo successfully produced the GMP resupply batch at full scale. Both drug substance and drug product was high quality, they met the release criteria, and the final product was comparable to the previous lot and accepted by the regulatory agency for use. The production timeline was compared to the original 3 CDMO scenario. It's like 6 months faster, and robust in-house analytical capabilities and well-coordinated third-party testing resulted in efficient release times. So I put the release data here. You can see both DS and DP are very high quality, and the process recovery for drug substance was about 90%. Let's switch gears, and I'm going to talk about Objective 2, which is the development of cost-efficient process through raw materials evaluation. The challenge here is the cost of the goods for RNA process is very expensive, and the supply chain reliability has been a concern. For that reason, Albac Medicines was interested in identifying alternative suppliers of key raw materials for future programs. So Kudo evaluated based on the Albac Medicines production process, the cost of the key raw materials contribution and potential impact of the change on CQAs. In the chart on the right side, the y-axis shows the cost contribution, and then the right side, the x-axis is the impact of the product quality. For example, T7 and cap analog, they are very expensive reagents. Switching T7 may have very high impact on the product quality. But something like NTPs, for example, this is not very expensive, and as they are chemicals, the impact of the change is not that significant. So based on this analysis, we decided to evaluate the T7 cap analog BSPQ1. This is the restriction enzyme to digest linearize the template DNA and some lipids. Supply chain team and quality team screen the suppliers to confirm the products meet the GMP requirement, and our MSAT team systematically evaluated 5 suppliers for each reagent using 2 different constructs provided by Arvac Medicines. In this slide, as an example, I'm going to share the results of T7 RNA polymerase screening. We tested 5 different T7 RNA polymerases and assessed the IVT products for the RNA yield, integrity, and dsRNA content. The plot on the left shows the IVT yield on the y-axis, RNA integrity on x-axis, and the color of each dot indicates the dsRNA level in each sample. Supplier A is the original supplier as a benchmark, and other suppliers' enzymes were tested in 2 different conditions. First one is Kudo's platform condition. It's labeled in black, and we also tested the enzymes in supplier's recommended condition, so it's marked in red. As you can see here, supplier A's enzyme performs the best in yield and quality, integrity. But also supplier C was identified as the best alternative based on the high integrity and low dsRNA content, so they were as good as supplier A's material. The yield was a little bit lower, but this is something we can optimize in the future with each construct with another DOE type study. Then we calculated the raw materials cost per yield, switching to supplier C's raw material, supplier C's T7 polymerase. It will reduce the cost of goods already by 40%. In addition to identifying cost-effective raw materials, you can reduce the cost by optimizing IVT conditions. So this is a result of another DOE study we performed for Arvac Medicine's next project, RVM V004. This is another linear RNA product with 2000 nucleotides long. So it's significantly shorter than their first COVID vaccine. We did the IVT optimization DOE study and identified good conditions like number 6 and 8, high yield and high quality. But among them, we identified this number 8 to proceed because this one requires 60% less T7 RNA polymerase compared to the previous condition. So we performed similar evaluation for other 4 critical raw materials like the BSBQ1, cap analog, and lipids. And then combining all of the findings, eventually the cost of goods was reduced by 68%. In summary, we achieved both objectives for Arvac Medicines. Our efficient GMP production and tech transfer delivered the clinical material with high quality in unbeatable timeline. We evaluated alternative suppliers for Arvac Medicines. We identified the raw materials suitable for their process and developed cost-effective process for their future programs. So here is the feedback from our client, Vice President of CMC at Arvac Medicines. Now I'd like to introduce our new Process Science Center of Excellence located in the Boston area. Our new site is located in Needham, and the team will focus on process development and also analytical development. All the work I presented today in the case study was done in our Shanghai site, but now with the opening of the Needham facility, it will be more convenient for our US-based clients. As listed in this slide, Our process science lab in Needham has similar capabilities to Shanghai site, but with more flexible scale, we can offer different services such as small-scale research material production and early-stage process and assay development work. Here are the standard QC assays we perform for DNA, RNA, and LNP drug product. Most of the assays will be done in-house, and we can help your product-specific assay development if needed. I have 2 summary slides to conclude this webinar. This is the highlights of our capabilities. So in this case study, we demonstrated our efficient tech transfer capability, process customization through DOE approach, alternative sourcing for high-value raw materials. We provide detailed batch records and study reports to our clients. And we are one-stop shop RNA-focused CDMO, so we have streamlined approach for your project needs, and we have very robust analytical capabilities. This is all with high-quality standards, and we can support the dossier preparation in both English and Chinese. Here's the take-home messages. Kudo is an end-to-end RNA-focused CDMO, so we can do all the processes from DNA to fill finish under one roof, all at one facility. We have extensive analytical and quality teams. They are very familiar with global regulatory requirements. We have exciting collaborations with various companies, including high-quality raw material suppliers and DNA technology companies, so we can offer various options for our client. Lastly, our new Needham site will provide support to our clients' process and analytical development needs with great flexibility, different scales. So if you have any questions, technical or business, please feel free to reach out to us. Here's the contact information for me and Kelvin. Thank you for your attention, and we are happy to answer any questions.
Thank you both Atsuko and Kelvin. We've had a few questions that have already come in during the presentation, so we can start with these. If we don't have time to finish all of our questions, we will reply to your email with an answer after the presentation. So it looks like the first question we have says, do you offer standalone services? Kelvin, maybe?
Thanks, Kathleen. Yeah, hi everyone. Hi, I'm Kelvin. I can take that question. I think the short answer is yes. We can and we do offer standalone services. As mentioned in the presentation, our process train is modular, and we offer through our partners with PacGene the pDNA, mRNA, LNP, and fill finish, as well as with analytical development and process development. Any support services as part of this workflow, whether it's cell banking, stability release testing, together with PacGene, we do offer all of these. And I think I also like to take the opportunity here to give a shout out to our fantastic partners at PacGene for collaborating with us on this webinar.
That's great, thank you. Okay, next question we have, it says, for the raw materials, did the company select the alternative suppliers that Kudo suggested?
Yeah, the short answer is yes. Can you hear me?
Yes.
Okay, yeah, so they chose the suppliers we evaluated and then identified for the future programs.
Okay. Okay, great. We have another question here. It says, where was this clinical trial held? Would Kudo's GMP production meet FDA EU requirements? Kelvin, maybe?
Thanks, Kathleen. I'll take that. Yeah, the clinical trial was held in Australia and in Singapore. So it met the Australian TGA and the Singapore Health Sciences Authority, HSA regulatory requirements. In general, our facility and the process is spec'd out for US FDA as well as EU requirements. So we are regulatory complying with the major global regulatory requirements.
Okay, good. Thank you. Next question. Are there considerations of exporting clinical trial materials cross borders?
I'll take that. We do ship around the world, and we have experience, shipping to the US, and I think if there are preferred couriers, shipping couriers, we are happy to work with you, especially if that's one of your choice. But we have experience of shipping them and exporting them around the world, particularly to the US too.
Great, thank you. Okay, let's see. Keep these questions coming, guys. What method was used to measure dsRNA levels?
Okay, so we have basically two methods to measure the dsRNA level. And then one is dot plot. It's semi-quantitative, but it's fit for purpose for many process work and stuff. In this case study I presented, we used the validated, more quantitative method. It's J2 antibody-based ELISA.
Okay, great. Do we have any more questions in the queue here? Let me see. Any more questions out there?
I see one in the chat. It's related to dsRNA, so I can take that.
Okay.
So the question is, how do we control or limit the dsRNA level in the product? So it's a great question. So in general, we do a DOE style optimization study I just presented in the case study, because it's better to limit the level of dsRNA during the IVT process. And then we have the purification process. It further reduces the contamination, dsRNA impurity. But we recommend to do some a PD work before we go, instead of going straight to the production, to confirm and then reduce the dsRNA level by optimizing your IVT conditions.
Okay, great, thanks. See if we have another question up here in the queue. Okay, does your LNP mRNA need 80-degree transportation system?
I can answer that. I think that was a minus 80-degree transport. Yes, we do ship it with dry ice, and the logistics coordinators that are companies that we work with have experience in doing that. So it is shipped under dry ice, end to end. Yeah. And I think there's temperature control systems put in place to make sure that there's control of variability. Any variabilities are reported.
Okay, great. Thank you for answering that, Kelvin. Let's see. Just got another one here. Okay, next question says, is the LNP packaging subject to any background IP and needs licensing?
Yeah, I can take that. So we are a technology agnostic company and a platform agnostic company. So we leave the IP licensing to our clients to coordinate for any of the lipids or any IP-laden considerations for the reagents. So that would be on our clients to work with these technology providers to get the IP. And yes, you would need licensing from them. If there are raw materials, it could be drop shipped to us, and we have experience working with the lipids in question here. I hope that answers your questions.
That was good. Any other questions in here? I don't.
Yeah, I think we see a question on formulation services for LNP delivery systems. We do offer those services for sterile aseptic liquids and lyophilized products.
Okay. Great. Any more questions out there?
I see that maybe it's like already in Kelvin's presentation, but what's your largest batch size and the limiting factor for the batch size?
Okay.
Kelvin, can you repeat the batch size?
Yeah, the batch sizes, let's say for GMP levels, at pDNA, we do up to 200 liters. mRNA up to 2 liters. Currently, the glass reaction vessel is at a 2-liter scale. But if there is a request to go up to 5 liters or more, we're happy to work to work with our clients and try to be as flexible as possible to see how we can, obtain such a larger reaction vessel. The alternative is to do multiple batches and combine it into a larger batch for bulk shipping or fill finish from there. At LNP scale, the batch size is pretty similar to mRNA scale. Yeah.
Okay, great. Thanks, Kelvin. Okay, any more questions here? Is that about it?
Well, I think, there are maybe a few more. So one is like, do you have some LNP RNA for oral delivery system? So this is like, we don't have it as a platform, but I understand like many people are interested in targeted delivery of LNPs, like by conjugating something or like using different lipids. So this is like a project basis, we can offer like development service with your need, but we don't have it as the established platform here.
Okay. Thanks for answering that, Atsuko. Appreciate it. Kelvin, any others that you have that come across in your?
Not that I can see.
No. Okay. Well, if we don't get any more questions here, I would just like to also thank you all for coming, for attending. Thank our speakers today and all the participants. And we hope you enjoyed your presentation and learned a lot about Kudo and Kudo Bio's capabilities and our CMC needs. Don't forget, we have a survey that you can take in the under the resources tab, where you're eligible to win an Amazon gift card, and it'll be perfect for the holidays. And we hope that you've enjoyed this, and we look forward to seeing you again at our next webinar. So thank you again. And if you've come up with any other questions that you'd like us to answer, please feel free to email us individually or reach out to us at info@kudobio.com. Thank you.